It is indeed a pleasure to introduce this afternoon's keynote speaker, Robert Plomin, someone so deservedly internationally recognized and well known as to truly not need an introduction, but I'm going to take a minute anyway. Robert Plomin's CV lists over 700 publications in dozens of different top journals, his work covering a vast array of different topics. He's also given hundreds of conference presentations in countries around the world, and as is the case today, he's been a sought-after keynote and invited address speaker. Following academic positions at the Institute of Behavioral Genetics and Penn State University, Robert came to the Institute of Psychiatry, King's College London in 1994, where he's currently Professor of Behavioral Genetics and Director of the Social, Genetic and Developmental Psychiatry Center. In 1994, he also launched the Twins Early Development Study, a study of all twins born in England and Wales in 1994 to 96. During the past decade, Robert's research has increasingly turned towards harnessing the power of molecular genetics, especially genome-wide association strategies, to identify genes for different psychological traits. For his many contributions, Robert has received numerous awards and honors, including being the youngest president of the Behavioral Genetics Association and a fellow of several academies. The title of his talk this afternoon, Quantitative Genetics in the Era of Molecular Genetics, and without further ado, please join me in welcoming Robert Plomin.
Thank you very much, Tony. And thanks so much to Tomas for organizing such a wonderful meeting. It's actually difficult giving a talk, a plenary talk at ISSID because it's such a broad church and people have such diverse perspectives. I'm pleased to see so much genetics here, but I thought for this talk, given that it is a plenary talk, I wouldn't talk to the geneticists here. I'd talk to the rest of people. So I hope that was the right decision to make. I'm going to be talking about quantitative genetics, by which I mean twin and adoption studies, and in the era of molecular genetics, by which I'm referring to DNA analysis. And as I mentioned this morning, I'm going to use Aesop's fable of the tortoise and the hare as kind of a theme for this talk. Quantitative genetics being the slow plodding tortoise, and the hare being molecular genetics. So you'll get more of that as we go along. These two worlds of genetics I find interesting. As you get older, you're interested in history, and I know nobody here under 30 is, but it still is quite interesting to think about these two worlds of genetics beginning over a century ago. The origins of, oh, I see, this isn't working really unless I talk like that. Is that right? Put it on the other side? Yeah, very good. Very good, except. Okay, is that better when I do that? Yeah? Okay. Well, it began over a century ago with the biometricians, so-called, and the Mendelians, these two worlds of genetics. And half a century before that, it began with Francis Galton, quantitative genetics, and Gregor Mendel, the ancestry of molecular genetics. What I think is really amazing is that, you all know about Mendel and the fact that his success had to do with identifying 7 single gene characteristics in the pea plant. And they're really like disorders. I mean, if you're a pea plant, having wrinkled seeds isn't too cool. So that's like a single gene disorder in pea plants. And as a result of that, he was able, for these qualitative dichotomous disorders, able to work out the laws of heredity. Now, at the same time, much less well known, Galton here, and Mendel was working with the edible pea. And what I think is really such a delicious irony is that Galton, being a gentleman of science, wasn't concerned about something useful like edible peas. He studied something that smelled nice, and it was the sweet pea, no less, And instead of a qualitative trait, because he was a breeding sort of person, he thought, well, everything's quantitative, so what would be a good quantitative trait? And so he studied diameter of pea seed size. And from that, he worked out the first regression that was ever published. And his student, Carl Pearson, developed the Pearson correlation coefficient after that. But he showed parent-offspring resemblance for a quantitative trait. And he and his followers believed that, they didn't even know about Mendel, but later on, as Mendel was rediscovered in the early 1900s, people went around looking for those 3-to-1 segregation ratios, whereas the biometricians were just laughing. I mean, they said, you know, this must be some quirk in the pea plant, because everything important in animal husbandry, plant, agriculture, humans, is quantitatively distributed. So what's this stuff about single genes and Mendelianism? So there was a vicious argument that went on for about 10 years between biometricians and Mendelians. And it's because the perspectives were so different, and they stay with us today in the origins of the differences between molecular and quantitative genetics. Not only is it focused on single genes and qualitative traits, it's also gene-centered. You know, molecular geneticists are interested in understanding how genes work. And so it's okay to feed Drosophila, X-ray them, or feed mice mutagens, and create experimentally induced genetic variation, because we want to understand how the gene works. In contrast, biometricians, quantitative geneticists, always thought that there were multiple genes involved, and that therefore they were dealing with quantitative traits. It's a phenotype-centered approach. In animal husbandry, agriculture, humans, we're interested in a phenotype, and we use genetics the best we can to help us understand that. And instead of inducing variation, you say, well, there's so much heritability and genetic variation already, we don't need to induce it. Let's study naturally occurring genetic variation. So those different perspectives really led the two fields to get farther and farther apart, even though by 1918, Ronald Fisher showed the resolution to this debate. You know, the debate being, was Mendel's laws of heredity just nothing to do with anything other than the pea plant? Or is there some way of reconciling Mendelianism with quantitative traits? And Mendel, R.A. Fisher's paper is one of those papers that nobody can read because it's quite opaque, but it's still considered the foundation of quantitative genetics. And he basically says Mendel's laws of inheritance for qualitative traits apply to quantitative traits if they are influenced by several genes. That's all it takes. So that if you just have one gene with additive effects, you know, 3 genotypes with 2 alleles, and then if you have 2 genes, you have 5 genotypes. These are with equal and additive effects, then 3 genes, 7 genotypes. But by the time you throw in a little non-additive genetic variance, a little unequal effects, a little environment, you get something that's indistinguishable from a normal quantitative distribution. So that was essentially what Fisher said. Nonetheless, the two fields diverged during the century. And when the new DNA markers were found in the 70s and 80s, molecular geneticists used them to try and identify single genes through linkage analysis by focusing on candidate genes. Quantitative geneticists were kind of out of it then because they were saying, well, there's so many genes, it's such small effect, you know, it's not really worth it. But these two areas, two worlds of genetics have come together sharply in the last decade, and especially because of these new DNA markers. Up till 1985, there were only a few dozen markers, genetic markers, like blood markers, which aren't genetic markers, they're just phenotypes that happen to be highly genetic. The new DNA markers were markers in DNA itself. So the one I'll talk about today is the SNP, single nucleotide polymorphism, where out of the 3 billion base pairs of DNA, there's one base pair that differs, say, between you and me. So that's a SNP. But we should, this is a good time to just remind you that 99% of our DNA sequence is identical for everybody in this room. And the 1% that does vary is what we're talking about with genetics. And there are millions of these SNPs, and there are many other sorts of DNA variants as well. So single nucleotide polymorphism, otherwise known as SNP. So those SNPs and the availability of millions of these DNA markers has led to kind of the revolution in molecular genetics called genome-wide association studies. And just to step back now and think about how much genetics has advanced in the last century. Yeah, it's interesting to think, just it was 1903 that the word was coined, genes, and it took 50 years for Crick and Watson to talk about the structure of DNA, then just 50 years till the whole genome sequence was known, mostly. But then in 2006, the first genome-wide association studies were published. But now we're already talking about sequencing the entire genome. And you know, if you think about here to there 50 years, then 50 years, it's just amazing and mind-boggling to think of what genetics will do. So we all really need to know about genetics. I was really struck with this because I wrote what I think is the first paper in developmental psychology on molecular genetics in Child Development in '98. And in a special issue on genetics and genomics coming out later this year, I think, I wrote a 13-year follow-up to that. And what was amazing to see is that what I was wittering on about in molecular genetics in 1998, the big things weren't even known then. So microarrays weren't even known then, which I'll talk about. That's the essence of genome-wide association. The whole method, in 1996 someone wrote a paper saying, you know, I think it might be possible sometime to do genome-wide association. And whole genome sequencing, it was only in 2003 that the human genome sequence was discovered. The idea that we would all have our full genome sequence of 3 billion base pairs, was just ridiculous. So genome-wide association then involves using hundreds of thousands or millions of SNPs across the genome to have an atheoretical, hypothesis-free approach to finding genes associated with complex traits. And you just get enough SNPs spread out throughout the genome, and one SNP correlates with the adjacent SNPs, 0.95, 0.99, so that no more SNPs would add much in the way of information. So we thought. So this is a GWAS, as it's called, genome-wide association SNP microarray. A microarray is like a slide the size of a postage stamp that has millions of probes, DNA probes. So a probe would be like a short sequence of DNA. And all it involves, it's the standard way of detecting polymorphisms, where we make your DNA single-stranded. So say this is your single-stranded DNA. On the chip, we have these probes, and we hybridize your single-stranded DNA to the single-stranded probe. And if there's a match, like the match has to be exact. So C wants to find G. So if you had a G there, you would hybridize. And we've attached to this, your strand, a fluorescence tag. So all we're doing is seeing if you hybridize with a probe. And if you do, then there'll be a fluorescence tag. And we have multiple markers of that probe so that we can see to what extent your one strand of DNA hybridizes with the probe. So, okay, that was just a method, but it paid off incredibly big, very quickly, with a gene being found for macular degeneration in the very first sort of published GWAS study, in a case control study. And it was a very big effect, an odds ratio of 5. The risk allele was, to give you an idea of what that means, 72% in cases. This is the wet sort of macular degeneration, 30 age-related, 36% in control, so double the frequency. And the next thing was everybody went out and tried to replicate it, and they did. It replicated because it's such a big effect. Then they found a second rather large gene where these two genes account for most of the genetic variants of the heritability of macular degeneration. The other thing that made it such a mind-boggler is both of these were in genes that nobody had ever thought of as being related to macular degeneration, but it was because they were involved in inflammatory pathways. And then it made good sense why inflammatory pathways were important, and so now there are a number of clinical trials with anti-inflammatory drugs. So I mean, this is what made GWAS. But it hasn't happened again. That's the problem. I mean, it's just amazing. With 90 families, they got this huge result, and nobody subsequently has anything like this. So that was macular degeneration. Then Crohn's disease, there were a number of SNPs found in genome-wide association studies, very much smaller effects, prostate cancer as well. Then this is what, was the classic study, the Wellcome Trust Case Control Consortium, 14,000 cases of 7 common diseases. So the 7 most common diseases, which I'll show you in a minute, each with 2,000 cases and 3,000 common controls. To do that sort of thing, you need collaboration. So there were 50 research teams and 240 authors. Here's what your author list looks like if you publish a paper like that. And the thing was, they also had success. So they found 20 associations reached genome-wide significance. Now, this is a Manhattan plot, it's called. These are the 23 chromosomes, and these are negative log p-values, so that this is a hit with a p-value there. But you can see there are a number of hits throughout. These are on the MHC, major histocompatibility locus. These genes in that region are associated with many diseases, but these were found as part of the GWAS study. So it was successful, and that really motivated people to do these GWAS studies. And because of that paper, it was named by Science as the breakthrough of the year. And it really has been successful. And this is a slide, the most recent survey of over 1,000 published studies with genome-wide significance for 210 traits. So here are 210 traits with significant associations across the genome. Here they are, the 210 traits, but here are the ones that might interest you. So it is making progress. It is being successful, but although people call it a Manhattan plot, I know people involved in that study said, whoa, there isn't a lot there. I mean, they were expecting lots and lots of hits, but there are actually relatively few. I think I call it a Brooklyn plot, because, you know, it isn't like you got all these skyscrapers. You got a few, you know, kind of tall buildings. And the odds ratio of this biggest effect, remember I said it was like 5 for macular degeneration? 1.4, and that's the biggest effect. So what we've learned is that the largest effect sizes are very small, and I'm guaranteed there's no one who would have predicted the effect sizes were all so small. They expected a distribution of effect sizes, but they expected the largest effect sizes to be bigger than that. And the largest one I know of, and please correct me if I'm wrong, because you know there's so many studies here. It's FTO is a gene that has been associated with BMI, body weight, body mass index, and it has about a 1% effect size in about 25 studies. So that looks pretty solid. And people 10 years ago, 5 years ago would have said, well, why bother with that 1% effect size? That, I think, is the biggest effect size. Height, you know, it's certainly less than 0.5%, and there are studies now in hundreds of thousands there. And the odds ratio for schizophrenia for the biggest effects is 1.1. So these are incredibly small effects. What people don't recognize enough is that these studies may be underpowered to detect effects of that magnitude, but they're tremendously powered to detect any bigger effects, even over 1% effect size, and they don't. So I think they've provided very strong evidence that there are no larger effect sizes. In my own studies, I did the first GWAS studies of reading, math, and IQ using multi-stage design, and we were powered to detect 1% effect size, which seemed really terrific at that time. Linkage could only detect like 10% effect sizes. So it sounded good, but our largest effect sizes are 0.5% of variance. And these are, you know, replicated effect sizes, but still, they're under the power, under the radar of the power we have to detect them, which isn't a good situation, right? So the question is, will they replicate? So I think most people have come around to realizing that what we're looking for are many genes of very small effect, which I'm going to call QTLs, quantitative trait loci, for reasons I'll explain in a minute. Okay, so we're not looking for major needles in the haystack, but for lots of little needles. I do want to just step back for a minute and say, yes, there are single genes. There's this neat kind of review in genetics that says there are 282 single gene disorders with monogenic effects and affect IQ. So you know about Fragile X and you know about phenylketonuria. But you may not know that neurofibromatosis, for example, has quite a big average IQ decrement of 1 standard deviation. So there are hundreds of these single gene disorders, but they're all rare and they're all quite severe. Look at the frequency of these. So that I think when you put this together in the population, they're explaining less than 1% of the heritability. So most of the heritability is not going to be explained by those. They're certainly important. I mean, if you had a child who had PKU, I mean, these are really important, but they're not going to explain most of the heritability of these traits. So this is the issue of missing heritability that was discussed a couple of times this morning. Heritability of most traits, personality, whatever, 40, 60%. But the largest GWAS associations are less than 1%. So even putting all the known SNP associations together, we're not explaining much variance. For height, for example, in a recent paper for 183,000 individuals, 180 variants were put together in a polygenic predictor and explained about 10% of the variance. But even that has to be replicated, and it's going to go down when you do more and more studies. So this gap between GWAS-identified associations and heritability is what's called the missing heritability problem. It's done more good for behavioral genetics, having a problem in molecular genetics named after something from quantitative genetics, heritability. I'm not joking either. It's really got people paying attention to heritability. So lots of things that people are thinking about to try and find the missing heritability. Obviously, larger samples, you all know that. Increase the power to detect smaller effects, so there are more and more collaborations and meta-analyses. You heard about some of those. Also, studying more extreme samples, you do get a lot more power if you go more extreme for a quantitative trait. Don't use Unselected controls use low versus high. You get a lot more power that way. And then other types of variants, like copy number variants. I'm sure you've heard about long sequences of DNA that are duplicated or deleted, not picked up completely by SNPs. The big one I think people are talking about is rare variants. On these chips, these microarrays, because they're more informative, they were composed of more common SNPs, like at least 1% frequencies, most of them over 5% frequencies. So people are saying, all we've shown with the GWAS is that common variants don't explain much of the heritability. Maybe rare variants would. So this is the big argument right now between the common SNPs that are on the current chips. They could detect effects that are small, and they haven't done so well there. Other people are saying, no, it's probably going to be more like single gene traits that are just specific to a family or an individual that you really have to detect by linkage. But I think, as usual with these arguments, it's probably all of these that are important. And that most of them, I think a lot of missing heritability will be found in these SNPs that are not as common as 1%, but they're not as rare as unique. And so chips are being developed here, so I hold out some promise for that. We know now, not only is this not it's highly unlikely, it's not the case, because I think GWAS has proven that there are no common SNPs with large effects. But here's the one that scares me that I don't think people are talking about enough. Maybe the effects involve rarish sort of alleles with very small effects. Let's just hope not. So there are other things people have talked about, functional polymorphisms. We've heard a lot at this meeting about candidate genes that are functional. Functional genes give you more power in these analyses. Other people are doing work on non-synonymous SNPs, if you know what I mean. These are, I won't bother to go into that. And they're defining functional polymorphisms by gene expression profiling using chips as well. So you can say, is this SNP functional? Not just is it a non-synonymous SNP that codes for a different amino acid, or that we've done the biological work showing it. You can do genome-wide sort of functionality by looking at SNPs against gene expression profiles across the whole genome. And you say, okay, if this SNP has a reliable effect on gene expression, then I'm calling it functional. So that's happening as well. With SNPs, people aren't just looking for genes now. Half of the replicated GWAS hits are outside of coding regions. Only 2% of DNA is coding, where it's transcribed and translated into amino acid sequences. But much of the genome, at least half of the genome, is transcribed but not translated. So it's transcribed into RNA, but the RNA isn't translated into amino acid sequences. And half of the known hits from GWAS are in what's called the dark region, or non-coding RNA, or in these regions that are transcribed but not translated. And then, as you see here, people are also looking at endophenotypes. I'm not as optimistic about it, but I say, you know, let 1,000 flowers bloom. Gene-gene and gene-environment interactions. So there's a lot of work being done trying to solve the missing heritability problem. Many of them, though, are kind of marking time, waiting for whole genome sequencing. So I thought I would also like to just tell you about whole genome sequencing. As I said before, the Human Genome Project was announced as, you know, finishing the genome sequence in 2003. It still isn't finished. But it took 2,000 researchers over 5 years and $3 billion. These are government statistics to do that, one, you know, sequence. And now, with high throughput factories with lots of machines doing it, you really can sequence your genome in one day for certainly less than 20,000. But there's this prize, you may have heard about the Archon prize of $10 million for the first one to develop a machine that can be handled by one person that can do 100 human genomes in 10 days for less than $10,000. And there's 6 groups very close to that. And if you saw Nature this week, there's a whole new technology that's just come out that people are very excited about, using semiconductors. So instead of those dyes, you know, fluorescent dyes I mentioned, this is actually detecting ion changes when you add one nucleotide that has, you know, different number of protons. And this device is being used now for 50 grand rather than 300,000 grand for what we've been doing up to now. And it can produce a genome not for $10,000, but for $1,000. But it still isn't, you know, rolled out yet, so we don't know how true that is. But these next generation sequencing techniques are certainly going to make it available to us, you know, to be able to do sequencing on a regular basis. For a few years now, there's been the 1,000 Genome Project. Wellcome funded a 10,000 Genomes Project. There are tens of thousands of other people whose genome is being sequenced. And once you get the sequence, it's sort of the end of the story. That's all there is in terms of heritability, from my point of view, which refers to inherited DNA sequence variation. It includes the non-coding genes, for example. And it's going to lead to personalized medicine. I really recommend Francis Collins' book called Personalized Medicine. And in there he says he's almost certain that complete genome sequencing will become part of newborn screening in the next few years. And if you don't know who Francis Collins is, he was the one who headed up the Human Genome Project, and he's now the director of the US National Institutes of Health. So this isn't just some crackpot saying this. And nobody's really noticed, but the important thing is, what if all newborns, and all of us say, have our whole genome sequence? Well, it means that we don't have to do any. We don't have to get DNA. We don't have to do genotyping. It's all there. So I think that's going to have tremendous impact. So will it be useful? I think without a doubt. We're going to learn a tremendous amount. In fact, from the human genome sequence, a lot of what we thought we knew, we found out we don't know. I mean, we mostly found out how little we knew and how much there is to know, like non-coding RNA, you know, the dark matter of DNA, the 98% that isn't in traditional genes. But will it solve the missing heritability problem? I bet a lot of money it doesn't, because so far when people get 3 billion base pairs, which an error rate of less than 0.1%, but that's 1 out of 1,000, and you've got 3 billion base pairs. So there are huge problems to even working with whole genome sequencing, but it's going to definitely be useful. And I don't think it's going to solve the missing heritability problem, because this is it, really. We're dealing with very, very small effects in many, many genes. But one thing that is happening, you've heard a few talks about this, and I think this is definitely going to work. It's a question of how good it will work, and that is, don't think about one gene. Think about these SNPs and heritability as like a questionnaire, a scale on your questionnaire. And you wouldn't use one item. You don't expect one item to do much. You put them together in a scale. So what if you just put together the dozens or hundreds of SNPs that you think are related to your trait in some sort of SNP set or polygenic liability? So that's where it's going. And we did that in our, say, our GWAS of math. These are the 10 top replicated SNPs. Here's the variance, 0.5 down to 0.1. This is percent of variance, very small effects. But you can put them together if you know that you have a 0, 1, or 2. So you think of the allele associated with math, 0, 1, or 2. You can add those together and get a score from 0 to 20, right, for 10 SNPs. And what's neat is it is completely normally distributed, right? And when you take the SNP set now, not the individual SNPs, and you correlate it, in this case, with standardized math scores, you find a much stronger relationship. Instead of correlations of 0.07, you know, the summed correlation for the SNP set correlation, 0.17, which is highly significant, and the effect size, instead of being less than 0.5% of variance, is like getting on to 3%. Now we have to replicate that and all of that. But what is interesting is you begin to use it, even with such weak effects, and what you want is hundreds of SNPs that are related to math. Like you want a scale that has a lot of items on it, even if some of them aren't right. Even with this really weak relationship, you know, still at the, if you did a genotypic selection of the guys low in SNP set score versus high, there's a standard deviation difference between those groups in their math score. So I think it is potentially useful. And so you can think about having lots and lots of SNPs. They're, you know, the most frequently used web science thing in America is the 23andMe, which offers genetic tests of 116 genetic diseases and traits. And there's also a cardio chip that's been around for 2 years that has 100,000 or so SNPs that have been shown to be related to some aspects of cardiovascular functioning and disease. An immunochip has come out in the last few months of 220,000 SNPs that are related to immunological functioning. So you can imagine using those, because the inflammatory stuff seems to be related to everything. Last year there was an attempt to get a neurochip for brain-related diseases that didn't quite work, because you need a large investment initially. And so I'd like to see a cognitive chip for my work because it's really what I've been after. I haven't been wanting to spend 15 years finding genes. I wish, you know, could have had a dream one night where someone told me what the genes are associated with cognitive abilities, because I want to use those for diagnosis. So we're not diagnosing on the basis of symptoms, but rather etiology. That we can intervene in sort of a personalized way, like personalized learning. And it's really the only game in town for prediction. You know, we talk about brain stuff. Well, brain behavior correlations are just correlations, and I think, you know, direction of effects is a big issue. But DNA sequence variation that's correlated with behavior is causal in the sense that nothing changes the DNA sequence variation, which is what I mean by heritability. So it can't be beat as an early warning system. It's not going to lead to genetic engineering. There are many genes of small effect. It'll be behavioral and environmental engineering. So molecular genetics, very fast moving. I don't think there's ever been a field of science that's moved as fast. But what have we learned? I think we've learned something very important, and that is that we're talking about many, many genes of very small effect. Now we can do things with that. I think we will have chips that account for a significant, I bet a lot of money, significant, but how much of the heritability? How much of the heritability is still going to be missing? And I don't think anybody knows. I certainly don't know. But it will be useful in research, because just as I showed you with our lousy 10-SNP set for math, you know, you can begin to use it in terms of genotypic selection, say for zero imaging studies, where you can't study 10,000 individuals, but you can select genotypically. But I don't, unless it becomes quite predictive, you know, I don't know that it's going to be useful clinically. So, quantitative genetics now, the tortoise in Aesop's fable of the tortoise and the hare. I don't like to be defensive about these things, but there is kind of a need to defend quantitative genetics because there's so much glamour around molecular genetics. But the glamour is wearing off as people say, well, where have these billions of dollars gone? I mean, what have we got to show for it? On the one hand, I want to say, well, we have some things. You know, it's macular degeneration, brilliant. But everyone's disappointed at the yield from the first harvest of studies. So the stuff I'm going to talk about now, some of it's presented in this paper. And it's really to say, why do we need quantitative genetics in the era of molecular genetics? And we talk about three main reasons that I think are really important. When molecular genetics tells us there are many genes of very small effect, they might interact and be very complex. But quantitative genetics gets at the bottom line, the accumulative effect of genetic influence, regardless of the number, magnitude, or complexity of their effects. And secondly, molecular genetics doesn't touch the environment unless you bring in environmental measures. But per se, it doesn't do environment. Whereas quantitative genetics is as much a study of the environment as genetics. So I'll talk a little bit more about that in a minute. And most importantly, quantitative genetics is thought of as the rudimentary nature-nurture question. You know, is it heritable? Well, everything's heritable. So that's not a very interesting question to ask. But quantitative genetics can go beyond estimates of heritability. And I think some of the biggest findings in the behavioral sciences have come out of that. So you all know about the twin method. It's kind of interesting, even at that level. Yeah, everything's heritable, but some things are more heritable than others. And here's a nice party quiz. If you go to nerdy parties like I do, academic parties, ask people, take these common medical diseases, which one's most heritable? Well, I know what people would say. I've done this. And for classes as well. Parkinson's, why is it heritable? You know, it's neurological, it's the brain. Breast cancer, all you hear about genes for breast cancer. Epilepsy, it's just a mess. I mean, you know, lots of reasons why people have seizures. Ulcers, stress, that's not genetic. So can you see people that say that's most heritable, that's least heritable? And what's really neat about this little party game is the results are exactly the opposite. So epilepsy is one of the most highly heritable characteristics. If you look at MZ, DZ concordances, okay, so MZ's concordance 0.8, DZ concordance 0.4. Breast cancer is hardly heritable at all. The concordance, you know, your base rate for women in the room is maybe what? Getting on 10%. Fraternal twins maybe 11%. Identical twins maybe 12%. So identical twins are clones. So if you had a clone who had breast cancer, your risk is not much elevated among the rest, for the rest of the population. And 85% of the time, you will be discordant lifelong. So breast cancer is one of the least heritable things around. Most cancers aren't heritable. They involve DNA, but that's another story. I'm talking about vertically transmitted DNA sequence variation. There's a lot of DNA events that aren't heritable. De novo mutations, chromosomal sorts of things. And mutations caused by cancer. Okay, and ulcers is also highly heritable. Parkinson's, I don't know why, there are all these GWAS studies appearing on Parkinson's, but it's the only trait I've found anywhere that shows an MZ concordance less than the DZ. You know, it's not significant. Okay, and one thing other that's counterintuitive is you look at the average difference between the MZ and the DZ concordance and you say, well, medical diseases, those are really heritable. Another assumption, behavioral measures aren't. Behavioral measures are more heritable on average. Nobody knows why that is. I think it's because they're further downstream. But for all of these, it's important, like breast cancer, to recognize that 20 years ago we had to say, these data provide very strong evidence for the heritability of schizophrenia. Now, in psychiatry, schizophrenia is a genetic disease. And so now you have to say, well, wait a minute now, identical twins are only 50% concordant for schizophrenia. We have clones, so 50% of the time, they're discordant lifelong for schizophrenia. There's no way to explain that in terms of inherited DNA sequence variation. It has to be environmental. So it provides strong evidence for genetics, but also strong evidence for environment. It's not just diseases, as you know, most quantitative traits, also personality traits, school tests, cognitive tests, IQ, show strong genetic influence. And I don't think we need to argue about nature versus nurture anymore. I finally did a survey. I kept trying to get people to do this, but I finally did it, where we asked parents and teachers, how heritable do you think these things are? And on a 1 to something scale. And what we find is that over about 90% of the time, both teachers and parents say that these things are, nature is at least as important as nurture. So I think most people accept that. I'd like to do it in academia, because I don't think that would be true in sociology departments, for example. So this nature-nurture work has shown that genetic influence is significant and substantial for most behavioral traits. It's not interesting to do a study asking, is this trait heritable? You've got to ask more interesting questions. And heritability means DNA sequence variation. We could talk about gene expression, epigenetics if you want. It's different. Genetic research also provides the best evidence for the importance of the environment. And we should keep emphasizing that. And environmental means, this is a very restrictive definition of genetic. This is a very loose definition of environment. It's everything other than that. And then next, and this is the other part of my talk, is quantitative genetics can go beyond estimates of heritability. And what I want to do in the next 10 minutes or so is give you 5 examples of that. And just, these are things we've learned that I think are very important. There's lots of other things, like you may have gone to the earlier session, there are lots of other things that we've learned from behavioral genetics. But these are some biggies for me, and I like the idea that some of the biggest findings have to do with the environment, like most notably non-shared environment. We consistently show that most of the environmental influence for behavioral traits, well, it's important, first, but then second, it doesn't work the way most of our theories of socialization assumed it works. It's not shared by kids growing up in the same family. It's called non-shared. So we do all the quantitative genetics to separate nature from nurture in family resemblance. But it turns out genetics accounts for almost all the familial resemblance. The environment's important, but whatever it is, it's not contributing to resemblance. And that's what we mean by non-shared environment. And so if you think of the adoption design, it's easiest. It's a social experiment, unlike the twin method, which is like a biological experiment, that separates genetic and environmental influence by studying kids, say, who are adopted away from their biological relatives, so they're genetically related parents and offspring, and then those kids are adopted by someone, and these are the environmental parents. And this is a direct estimate of shared environment. So these are kids adopted early in life into these homes. And so with just taking weight as an example, here's parent-offspring results. Correlations of 0.25 for genetically related relatives, even when they're reared apart. Adopted parents and their kids don't correlate at all, 0.03 for weight. So there's no evidence for shared environment. Similarly, a third of adopted families adopt another child. They're genetically unrelated, environmentally related siblings. Again, they don't correlate at all for weight. Whereas regular siblings correlate, about 0.35. So if you put all the data together, this is what you get. The variance of weight, very substantially due to genetics, which a lot of people don't, understand. They talk about height, but not weight. Very little shared environment. And the important thing is our environmental theories of weight assume, implicitly at least, that the important environmental influences are shared by children growing up in the same family, like nutrition and lifestyle. But that really isn't the case. Something else, environmentally, is important. It's not just what I study, cognitive abilities, but psychopathology, personality. There's always exceptions to the rule, but the striking thing is how important non-shared environment is. And cognitive abilities after adolescence, which I'll come on to in a minute. In the recent issue, the current issue of International Journal of Epidemiology, I have a paper that summarizes 3 decades of research. Must be getting old. All my papers are like 13 years later, 20 years later, 30 years later. So that's a little worrying. The second example, heritability increases during development. These are the world's literature on quantitative genetic studies, family adoption, twin studies of IQ. Typical pattern, 50% heritability. Unlike most traits, a quarter of the variance due to shared environment. But if you look at the data, most of those studies have to do with children, and they show substantial shared environment. But you look at the studies after adolescence, and they don't show much shared environment at all. If you plot the literature, you get this of studies cross-sectionally by age. You find that the heritability increases linearly from about 20% in infancy to maybe 40, 50% in adolescence, and even higher later in life, excluding dementia. And what I like about behavioral genetics is, okay, there's a trend, cross studies or whatever, but several of us got together in a consortium that included 11,000 pairs of twins from 4 countries, and we wanted to just test this hypothesis. So here's the world's literature. Where do these data fit? What I like about behavioral genetics is the stuff replicates. Here are the results for that meta-analysis. You know, they're bang on the line. So I find that really impressive. Big question is why? I mean, this is counterintuitive. Again, at your nerdy party, if you ask people, do you think heritability increases or decreases over the lifespan? Once they understand what the heck you're talking about, they'll say environment increases, you know, slings and arrows of outrageous fortune. Heritability is what happens early in life, you know, all wrong. But most people think it must have something to do with GE correlation. That is, that initial genetic effects they snowball, they have bigger and bigger effects throughout life. But nobody's tested that I know of. The third example is generalist genes, and I mentioned that a bit this morning, so I'll go over it quickly though. It's an important advance in quantitative genetics, where you don't just look at the variance of one trait, say reading, then do a univariate study of another trait like mass. But you can look at the covariance between these traits in order to ask about the extent to which the genes that affect one trait also affect another trait. And that's the new construct of genetic correlation. And one of the findings that was most striking to me in, say, dealing with learning abilities and disabilities is if you look at the cognitive theories of reading and math, these are 2 completely different things. Yet the phenotypic correlation is substantial, but the genetic correlation between standard tests of reading and of math is 0.8. That means that if you found a gene for reading, this is a rough interpretation, there's an 80% chance that that gene would also be associated with math. And it's not just for reading and math. You get similar results for language, cognitive abilities in G, and other domains, like we heard this morning about psychopathology. So here's our most recent study where we have a latent trait approach to reading, math, and language, where we have multiple tests of each of those. And A is additive genetic variance. I'll just talk about that now. The genetic correlation between reading and math is 0.75, between math and language, 0.78, between reading and language, 0.91. Then you look at G, so going outside of these sort of school achievement-like measures to general cognitive ability, IQ, you find even higher genetic correlations, meaning that whatever IQ tests are assessing, they're kind of getting at an index of whatever's involved with generalist genes. I don't think it explains anything, though, for reasons I can go into a bit later. So Yulia and I published a paper in Cycle Non-Generalist Genes, and we've talked about implications for psychology, neuroscience, and molecular genetics. And I'll just give you this one, I'll just do it very quickly. When you say that, neuro people think of a single process that's responsible for it, and that in the brain or in the mind, any process will affect many behavioral traits, so that the generalist stuff comes about, not because there are generalist genes in the brain, but because different processes are involved. Most neuro people with a modular approach to neuroscience would think of this model, multiple independent processes, where there are processes in the brain or the mind, all of which affect most behavioral traits, creating covariance among them, but that these are genetically independent modules. But I'd be willing to bet a lot of money that's not true, because the 2 things that are important about genes and the way they work is pleiotropy any gene has multiple effects. And polygenic, any trait is affected by many genes. And I'm sure that's going to be true in the brain as well. So they aren't independent modules. Many genes affect many brain or mind processes as well. Okay, example 4, quantitative traits. When we're talking about heritability, we're usually at ISSID talking about individual differences heritability, individual differences in a population. But there is a method called DF, the Fries-Fulker extremes analysis, that gets at something else. It's called group heritability. And it's saying, at the extreme, or for whatever extreme group you want to select, to what extent do they as a group differ genetically and environmentally from the rest of the population? It's not dealing with individual differences within the group. It's saying, do the extremes differ genetically from the rest of the group? And you can think of it this way, you get your probands and you can look at their co-twins, whether they're MZ or DZ co-twins, and the extent to which the MZ and DZ co-twins regress back to the mean suggests it's not entirely genetic. If they were both at the population mean, it would mean it's all non-shared environment. This would suggest some shared environment and some heritability. But on the group heritability side, it's saying that the average difference between the probands and the population is substantially due to genetic differences. But it's talking about genetic links between the normal and the abnormal. It's not talking about individual differences within the group or within the population. And typically what you find is that group heritability, this is for reading, the individual differences heritability for the whole sample is quite high. It's true at the extremes of individual differences as well, but group heritability is also quite similar. So that suggests genetic links between the normal and the abnormal. But I wrote this paper for Nature Reviews Genetics, looking at it from a GWAS point of view, a molecular genetic point of view. But it's really going back to Fisher and this business about if you have multiple genes, if there are multiple genes, it would explain quantitative traits in terms of Mendel's laws. But what about the reverse? If you got a qualitative trait, like most GWAS studies are on disorders, case control studies, if it's influenced by several genes, the genetic liability is likely to be quantitatively distributed. Because I showed you any SNP set will be quantitatively distributed. So the idea of the QTL hypothesis is that what we call disorders are the quantitative extremes of the same genetic and environmental factors responsible for normal variation. So it's important to study extremes like kids who have reading problems, for example, but it's also important to realize that what we're dealing with is a quantitative trait. And common disorders are quantitative traits, or more polemically, there are no disorders. There are only quantitative traits. We focus, like using polygenic risk scores now, on the problem end. But I showed you that they're normally distributed, and nobody studies the other end of this distribution. And so I'm interested in talking about not positive psychology, I don't know if you're sick to death of it, but positive genetics. Because anything that's here, that SNP set has the other end. And what's there? And you can talk about the ability instead of disability, enabling, resilience, that sort of thing. But I just like the idea, and it's not necessarily all positive. It may be evolutionarily what's important is in the middle, for example. So I think it's an interesting thing that's opening up positive genetics. And lastly, and just about 3 minutes, I think, we have a business meeting at 4, so I want to make sure we're done by then. I think this finding is really remarkable. It's been around now for 20 years, but it still hasn't had the impact I'd like it to have. And that is that not only are environmental measures heritable, but I think genes affect experience. And so the traditional model of environments is reasonably enough. An environmental measure measures the environment, and behavioral genetics talks about genetic and environmental influences on a behavioral phenotype. But what if you treat the environmental measure as a dependent measure? People would say, well, it's crazy, it's an environmental measure, how can it be genetic? But you say, well, okay, let's just look at it. And when you think about it, you realize that measures of the environment don't just involve the environment out there independent of us, they always involve our behavior. So in 1991, I published this brain and behavioral science paper that had 18 studies that looked at the heritability of environmental measures. And the first thing you see is most of them are heritable. So this dimension is from 0 to 50% heritability. But also, it's not just self-report measures. This scale, subjective to objective. Videotaped observations, SES, home observation, video, they all show heritability as well as self-report measures. It's also interesting, unlike behavior, there's stronger evidence for differential heritability. Parental warmth in several studies is more heritable than parental control. And it'd be interesting to talk about that. You might say, oh yeah, well, you know, how much parents love their kids has to do with how loving they are. But you can easily come up with counterexamples where it should be the other way around. If I had tricked you and said, this is more heritable than that, you'd come up with theories as to why that would be. So other ones, controllable life events versus uncontrollable, makes sense. Death of my spouse, I hope, doesn't have a lot to do with heritability. Illness and that sort of thing, financial disruption has more to do with me. I did this perverse study of television viewing just because it irritated me that people assume television viewing in 1,000 studies is viewed as an environmental factor. And it is one of the more heritable ones. So I'm now working on, again, another how many year follow-up of calling it genetics of experience. And there's now been, since 1991, more than 150 papers, and they confirm these sort of basic findings that I mentioned to you, and have some newer findings, boars, peer deviance, classroom environments. And very interesting, the most heritable of all is bullying victimization. It's not bullying, it's being victimized by a bully. The next step then is to say, well, if behavioral measures are heritable, environmental measures are heritable, maybe the relationship between environmental measures and behavioral measures are mediated genetically. And so about half of those 150 studies are doing that I mentioned, the new study since 1991. There wasn't a single study in 1991 that had done that. Genetic measures contribute substantially to measures of the environment, and genetics also contributes substantially to the association between measures of the environment and behavior. So that's the reason why we have to study the environment in genetically sensitive designs. It suggests that we move from a passive model of the environment, where the environment's what happens to you, to an active model of experience. And the idea is that people select, modify, construct, and reconstruct and perceive their experiences on the basis of their genetic propensities. Doesn't that sound good? Try and figure out what that means. Try and construct a measure to do that. I've been beating myself up trying to do that. So why quantitative genetics? Again, let me just emphasize these 3 reasons why quantitative genetics continues to be very important. And I talked about 5 examples of findings that I think are big findings in the behavioral sciences. And so in closing, I'd like to say that most of what we know about genetics and behavior comes from quantitative genetics. Molecular genetics is very important, but most of what we actually know comes from quantitative genetics. And I hope Tim Bates' talk this morning and what I've talked about convinces you that there's much more to learn from quantitative genetics beyond heritability. And finally, I hope you can now see what the moral of Aesop's fable is. Thank you very much. I'm told that there won't be questions now, but I'll be around if people want to ask. And we're moving